right, welcome back to virology. Today, we're going to talk about unusual infectious agents. These are things that don't fit in anywhere else, but are nevertheless really, really interesting. What is the minimum genome size needed to sustain an infectious agent? This is one question we're going to answer today. And then, could an infectious agent exist without a genome? And today we're going to talk about viroids, satellites, and prions to answer these questions. So let's start with viroids. So these are not viruses, they're viroids. That's why they have a different name. They're circular, single-stranded RNAs between 120 and 475 nucleotides. They're extensively base paired, so they appear as these rod-like structures when you look at them in the electron microscope. They don't code for any protein, yet they're infectious agents, as you will see here. So you don't need to have any protein to be infectious. They migrate from host to host. There's no receptors needed, and they replicate when they're introduced into plants. And there are two families, the POS-P Viroidae, they replicate in the nucleus of the cell, and then the Avson Viroidae, they replicate in chloroplasts, right? Plant cells have this other organelle called the chloroplast, and they reproduce there. So how does this work? So some of these viroids are what we call ribozymes. These are enzymes based in RNA. We'll look at this in a moment. There's no protein needed for the enzymatic activity. They're self-cleaving RNAs, and that activity for those viroids that have it is essential for their reproduction, or replication, I should say. And they're different from the virus. Viruses are parasites of the translational machinery, as you know. They all have to produce mRNA that can be translated by the host cell. But viroids are parasites of the transcription machinery. They don't need the translation machinery. No protein is made. Transcriptional machinery, as you will see, copies the viroid genome, and so they're parasites of that. So it's very different from viruses. The first viroid was discovered in 1967 by Theodor Diener, T.O. Diener, who just passed away in 2023 at 102. So if you want to live long, study viroids. Isn't that cause and effect? And potato spindle tuber viroid he discovered. It's kind of the prototype. It's been studied a lot. 359 nucleotides in length. And this one interferes with potatoes. And so when farmers plant potato seeds, they have to make sure they're free of this viroid. And there are laboratories that provide viroid-free seeds for farmers. There's one upstate New York in Ithaca at Cornell University, for example. Some of these viroids are benign. They don't cause any disease. They reproduce in the host, but the host is asymptomatic. But others cause disease, and this is economically important, right? So here's an example of, this is a tomato plant, and you have 3 different viroids infecting this tomato plant. You can see the one here, RG1, is causing extensive stunting. The tomato plant is not growing properly, and that's not good for a farmer, right? And then there are intermediate and mild symptom-causing viroids as well. So these are agriculturally important. As you'll see, they have to be taken care of because they interfere with production. Now, I have some favorite ones I want to share with you. The Kadang coconut viroid. They're favorites because of the names, okay? Kadang, what could be better? It causes lethal disease of coconut palms. So there's a coconut palm, and it's all brown, right? Because it has a viroid in it. And this makes pina colada drinkers sad, right? Because that drink has coconuts in it. Then there's the hop latent viroid. Hops are the plants used to make beer, right? This one has no symptoms in the hop plant. There's a hop plant infected. You can't tell. Beer lovers are happy. because it doesn't affect beer making. And then my third favorite is the apple scar skin viroid. This gives mild symptoms, like that apple has apple scar skin viroid infection. It's mottled. And if you saw this in a supermarket, you probably wouldn't buy it, right? You want a pristine apple. And so you can't have this because nobody will buy the apples. I would. I think it would be cool to eat an apple like that. But people say, what's wrong with it? I don't want that. So those are three of my favorite names. Viroid RNAs have functional regions. So at the top is potato, spindle, tuber viroid. So the names end up with VD, which is viroid. This is the viroidy. This has this rod-like shape. And typically there are left and right termini. There's a conserved region. And then there's a region that's essential for pathogenesis, for it to cause disease in the plant. We'll talk about what that does in a moment. And then at the bottom is peach latent mosaic viroid, PLMVD, which is an avson viroid. Remember, this one reproduces in the chloroplast. And you can see the structure is very different. It has a very different series of RNA stem loops. It's longer. And it also has a ribozyme right here. So this sequence is self-cleaving. It cuts itself. And that's part of the reproduction cycle. So it's actually an enzyme embedded in the RNA. It's not a protein. It's an RNA-based enzyme, which is basically a remnant of the RNA world, right? We think there was an RNA world at the beginning of Earth. And that RNA world got pretty involved. And one of the features was to have RNAs that could have activity like self-cleaving, self-replicating, and even protein synthesis eventually. So these viroids carry out both vertical and horizontal transmission in plants via pollen. So vertical is from the mother plant to the offspring, and horizontal, of course, is from plant to plant. And these are two diagrams of how that happens via pollen. So the pollen is infected. So the pollen is produced from a viroid-infected plant there, and the pollen contains the viroid. And so this can travel from plant to plant. So here's an example of a pollen granule going to another plant, and then it's taken into the pollen tube, and the viroid then invades the tissues. So that's one way that you get horizontal transmission. And then there's vertical transmission. The pollen infects the mother plant, as you see here on the right, and then the viroid ends up in all the tissues, including the ovule, which is the precursor of the seed. And then the seed ends up having viroids in it as well. So if you produce seeds for farmers to plant, you have to make sure they're free of viroids, because otherwise the crop will be defective and will lose a lot of money, and the farm may go out of business. So these are really agriculturally very important. Now, how they replicate, they're copied by the host RNA polymerase II. So you may, I don't think we talked about it yet, so you won't remember. So I won't tell you until we talk about it. So this is very interesting. It's RNA being copied by a cellular Pol2, which is a DNA-dependent RNA polymerase, right? It makes mRNA. Pol2 makes mRNA. Yet, in these cases, it can recognize those viroids and copy them. And one of these, this is the, I can't remember the name. This is the viroidy, right? This is the viroidy with the rod-like structure. This doesn't have a ribozyme, but the avson viroidae encode a ribozyme, which I just showed you, so it can cleave itself. And this ability of RNA to catalyze a reaction without protein was discovered in 1981, and it led to a Nobel Prize for the discoverers because it's very unusual. And during replication, as you see, you make multimeric structures that are going to be cleaved by this ribozyme activity. And the other viroids are cleaved by host nuclear enzymes. But we'll see that here. So here's the viroidae, which are the rod-like ones without a ribozyme. They enter cells. How they enter cells, we'll talk about in a moment. They enter cells, they go into the nucleus where they're copied by Pol2. And Pol2 makes concatemers, right? Multiple copies of the viroid genome all linked together, just showing 2 here for simplicity. These then go into the nucleolus, where they're cleaved and they circularized, and then they're exported and they move from cell to cell through these junctions in cells in plants called plasmodesmata. So this is a plant cell that we're showing here. Most of these, until very recently, were thought to just infect plants. And so a lot of study was done in plants. The other family, the avsun viroid, they're more structured and they encode a ribozyme. So these enter the cell and go to the chloroplast, where they're copied by a DNA polymerase of chloroplast. So in the previous example, this copying was done by nuclear Pol II. And here the chloroplast DNA polymerase is copying this and making RNA copies, which then are cleaved, they refold, they replicate again, and cleaved and so forth, and they move from cell to cell. And this cleavage, excuse me, this cleavage is carried out by the hammerhead ribozyme. So in the sequence of the viroid, there's that short sequence that is self-cleaving, and it cleaves itself, and that separates the 2 pieces. So that happens in the absence of a cell enzyme. So where did they come from? Where did viroids come from? Well, they were discovered in the 20th century, as you know, 1960s, because many of the plants we use for agriculture now were derived from wild plants, right? They didn't just magically appear. We derived them from wild plants. We made breeding lines, and those wild plants had viroids in them. So at some point, someone noticed symptoms in a potato plant, and it was linked to the viroid by T.O. Diener. So we think they're relics of an RNA world, as I said earlier. And probably the reason they're a problem is because we do monoculture. We use plant breeding lines that are identical globally. So there's just a few big companies that make the seeds and everyone uses them. It's called monoculture, and so that leads to having viroids in many different plants. So how do they spread? Largely by mechanical transmission in the field. Farm machinery can be contaminated, farm equipment, like shovels and so forth. Farmers' hands can have viroids on it, and they can move it from plant to plant. As far as we know, there aren't any vectors involved, but we could be wrong there. It's largely the farmers doing their manipulation that transmits these. So how do they cause disease? So there again, that picture of the different pathogenicity viroids. Basically, when this viroid RNA enters a plant, the plant recognizes it as foreign and chops it up into siRNAs, small interfering RNAs. Remember, many organisms have this system. When foreign RNA comes in, it's chopped up, and those small RNAs are then used to silence more incoming genomes, which is why they're called small interfering RNAs. Well, these RNAs, so the plant sees this as foreign, it chops it up into siRNAs, but those siRNAs also target plant genes, kind of an off-target effect, right? And that's what causes the disease in the plants. These viroid-derived small interfering RNAs, they cause silencing of plant mRNAs, and that leads to the disease. And you can show this by showing that the symptom development correlates with the production of these small interfering RNAs. And most of them map to this pathogenicity modulating domain here on the left that I originally pointed out. And it was found early on, if you make base changes in this pathogenesis region, you alter the pathogenicity, and that's because the siRNA are derived from that region of the viroid. Now, recently, people have been searching metatranscriptomes. What is a metatranscriptome? As you know, people now can take any cell and extract mRNA and sequence it. It's called a transcriptome because it's of many different cells, and its transcript is mRNA. And metatranscriptomes are from taking environmental samples like soil or water and just sequencing all the mRNA from it. So these sequence projects are deposited in public databases, and you can go search them. So people went in and searched using signatures of viroids. And they found that there are way more viroids than we realize. They're not just in plants. And so first of all, they're globally distributed. These are some of these new hits found in transcriptomes. So they're viroid-like closed circular covalently linked RNAs, just what I've shown you, these circular molecules, single-stranded. And they've been found throughout the world, on land and in oceans. And they seem to be associated with not just plants. So here's plant litter and plants themselves. So you can see there are plenty of them there, but there are also lots in the soil. There are lots in the oceans and fresh water, engineered samples like, like a dam or something in some environmental feature that's been engineered by humans. Aquatic and animals even have some in them. In fact, recent studies showed that the human intestine is full of these viroid-like RNAs. We didn't even know they were there before, but looking at these metatranscriptomes, they've been found. So they don't just apparently infect plants. They infect many other organisms. In humans, they've been called obelisks. You might have seen that reported in the press because they're rod-like and they look like an obelisk. What's an obelisk? Well, you think of the Washington Monument in DC, right? It's an obelisk. So these rod-like RNAs look like that. So much more to be learned in coming years about what these are doing. And another thing to think about, what's in you, besides all the viruses I've told you all have, now you're full of viroids in your intestines. We're back to the quiz again, you noticed. So here's when people complained and I returned. So today and next time we have quizzes. Which of the following is not a property of viroids? Some contain a ribozyme. They're replicated by host RNA polymerase. They have a metastable capsid. They're circular, rod-like, or none of the above, which is not a property of viroids. All right, let's have a look. So the answer is metastable capsid, of course. Viroids don't have a capsid. They're naked RNAs. But all the other answers, they contain a ribozyme. Some contain a ribozyme, that's right. They're replicated by host RNA polymerase. Not all of them, but some certainly are. They have circular rod-like genomes. But they do not have a capsid, and they don't encode any proteins whatsoever, so there can't be a capsid. Now let's talk about satellites, not the kind up in space, but these are virus satellites. And a satellite is a virus that depends on a helper for its replication, all right? So you could sort of think as Rouse sarcoma, well, Rouse sarcoma is not defective, but all the other transducing retroviruses that are defective, they require a helper, which is avian leukosis virus. So you could think of them as satellites. But these are viruses with single-stranded RNA or DNA or circular RNA genomes, and they lack the genes that they need to replicate. And there are two kinds that we recognize. We recognize satellite viruses. So these satellites encode structural proteins that make a capsid. So they make particles, and they're different from the helper virus. But they don't have replication genes, so they depend on a helper virus to provide, say, an RNA polymerase. Right? So that's a virus. And then we have satellite RNAs. These are packaged by helper virus proteins. So it sounds like there are naked RNAs, but they're not. But they are called satellite RNAs because they cannot encapsulate themselves. They're packaged by proteins provided by the helper virus, and they also rely on the helper for replication. These satellite RNAs may or may not encode protein, whereas the satellite viruses do encode structural proteins. All right, so that's what a satellite is. And let's look at some properties of them. On the left are how these are classified. Some of them are linear RNAs, and they have a single open reading frame, which encodes a non-structural protein, which means not a capsid. It's probably involved in reproduction. Then we have linear RNAs, do not encode protein. And then we have some circular RNAs that do not encode protein as well. So these are just three different categories of satellite RNAs. And the way they reproduce, they enter the cell. And remember, the helper virus is also infecting this cell, and it provides an RNA polymerase that will copy the genome. These are typically, for the 2 out of the 3 classes, they're linear. They become circularized on entry into the cell for the circular RNAs. You skip that step. And then they're copied by the helper virus polymerase, typically a rolling circle replication that makes concatemers that are then cleaved by nucleases and typically cell nucleases, and then you have your individual genomes produced. And here are some examples of satellite viruses. So one that we have talked about a bit is adeno-associated virus, AAV, which we'll talk about next time when we talk about vectors. It's a very popular vector for gene therapy. So the AAV genome is a single-stranded DNA. Remember, with the terminal T structures, it does encode a capsid protein, but it needs a helper virus to get it to replicate. And the helper can be either adenovirus or herpes virus. So it has to be, in order for this virus to reproduce, it has to be co-infected with an adenovirus or a herpes virus. So that's a vertebrate satellite virus. And then we have a satellite virus of bees. Chronic bee paralysis virus is the helper for CBPV satellite, chronic bee paralysis virus satellite. And this satellite, these are properties of the satellite here. It's a single-stranded RNA. You do get a particle. The genome is this long and it encodes a capsid protein. And then finally, there's a plant. There are many examples of these. This is just one. And then there's a plant satellite, tobacco necrosis virus satellite, also single-stranded RNA that encodes a capsid protein. And tobacco necrosis virus is the helper. So in these cases, the satellites encode the capsid proteins, but they can't be replicated without the helper virus. So here's an example of how this works. This is adeno-associated virus infection of cells. So this is a single-stranded DNA virus, attaches to the cell, the DNA genome gets in the nucleus. Remember, these T structures form, and it undergoes a replication scheme that we have talked about before. This will not occur, this reproduction scheme will not occur without a helper adenovirus. And among the different proteins that are needed are E1A and other early proteins that facilitate early steps in reproduction, in particular transcription. And so we know exactly how many adenovirus genes are needed for AAV. And so when we make vectors, as you'll see next time, we take advantage of that. Satellites, when they're present, cause the plants anyway, to have different diseases that aren't seen when the plant is infected with the helper virus by itself. And these include necrosis, which is destruction of the leaf, or chlorosis, which is when there's no chlorophyll present. So you see that plant has chlorosis. It's got yellow spots, which means there's no chlorophyll there. There's no green. And those are caused by the combination of helper plus satellite. Now, an important thing to remember is that these satellite viruses are not derived from the helper. Their genome has no homology with the helper virus. So it's a unique virus that probably at one point was autonomous, right? It could reproduce on its own. And then it lost genes, perhaps, but it had a helper around to enable it to reproduce, and that combination was evolutionarily selected for. So it is a distinct virus from the helper. There is a human satellite virus called hepatitis delta virus. And it's interesting because it has properties of both viroids and satellites. Its helper is hepatitis B virus. And it's thought that, hepatitis B virus causes hepatitis in humans, right? We talked about that before. It is thought that if you're co-infected with hepatitis B virus and hepatitis delta virus, you will have more severe liver disease. There's a little bit of controversy around that, but some people are co-infected with both, and we think they have more severe hepatitis as a consequence. About 18 million people globally are infected with hepatitis delta virus, HDV, and that's about 5% of the people who are infected with hepatitis B virus. And these are the parts of the world here where you can see the prevalence. You can see the highest regions, looks like Japan and parts of the Middle East, and then intermediate, mostly mostly Asian and Pacific countries. It used to be high in Europe, but it seems to be declining there recently. But many people are carrying both Hep B. And you can't have Hep Delta alone, right? You can only have Delta with Hep B. You can have hepatitis B virus alone, but you cannot have Delta alone. It will not reproduce. It needs a helper. So this is the genome of hepatitis delta virus. It's a 1.7 kb single-stranded RNA that's extensively base paired, so it appears like a rod. So that's the viroid-like property. And there is a genome and an antigenome present, and then there's a ribozyme sequence in it right here where it says self-cleavage. That's also reminiscent of a viroid, right? Because as these viroids, as these satellites replicate, they are self-cleaving. And it is a single mRNA produced from the genome. It's shown here, and that encodes two proteins, the small and large delta antigen, and that's going to be involved in the packaging of hepatitis delta into a virus particle. And so the relationship of hepatitis B and delta is shown here. Delta is copied by the host cell RNA polymerase II. I'll show you that in a minute. But what it gets from hepatitis B is the envelope glycoproteins. So Hep B is enveloped, and in the envelope are a variety of Hep B glycoproteins, and that is what is provided to hepatitis delta genome. So in this particle on the left, the delta genome is shown there, and the two delta antigens are shown. So the small delta coats the RNA, and then the large delta is thought to be embedded in the viral membrane, and that interacts with the RNA. So it's probably involved in packaging. So this is basically the hepatitis B virus envelope derived from Hep B that packages the delta genome. So this is a delta virus RNA genome in the envelope of a hepatitis B virus. The way this reproduces is it's copied by host RNA polymerase II. Same for some of the viroids. So what comes into the cell is actually a negative strand. So this RNA in the particle is negative strand, and this is the right color for negative strands, this kind of olive color here. So the negative strand comes in, and RNA pull 2 begins to copy it and makes a full-length copy, and then it keeps producing longer and longer concatemers, basically. We're only showing one here. This little red circle is the self-cleavage site, so the RNA cleaves itself, and in some cases an mRNA can be produced, but in other cases you make a full-length plus strand, and that circularizes, and then from that you make full-length minus strands, which then end up getting packaged into virus particles. But again, the self-cleaving activity, the red circle, is essential for reproduction, and all of this happens by RNA pull 2. And then these genomes, these delta genomes, are packaged into the hepatitis B virus particle. So the genome can reproduce in the absence of Hep B, but it will not package unless hepatitis B virus is present. Recently, by looking at RNA sequence data, delta-like viruses have been found in birds and snakes. And so they've been discovered in RNA transcriptomes of healthy birds and sick snakes without hepatitis B virus. There's no Hep B there. So maybe there's another helper, because these delta-like viruses can't be packaged on their own. Maybe they're not, but people are looking for a helper. And in fact, with hepatitis delta, if you put delta virus in cells in culture, other viruses can package the RNA, including hepatitis C virus, VSV, dengue virus, and West Nile virus. So that's for delta. And no one has done the experiment yet for these bird and snake delta-like viruses. But again, just like viroids, they're much wider than we anticipated. We thought delta was this human-specific pathogen, but it's not. Delta-like viruses are present in birds and snakes, and probably more. This is a phylogenetic tree of all of these delta-like viruses. So here are the human isolates up top here, and then the snake and the avian delta-like sequences are way off the beaten path. You see, they're phylogenetically very different. So they have they seem to have some common ancestor, but they've gone off on their own in terms of evolution, which is not surprising. Okay, next question is, which of the following statements is correct? There are no human satellites. They are defective. This is about satellites, of course. They are defective viruses derived from the helper. Like viroids, they do not encode protein. In plants, they cause symptoms distinct from the helper virus. What is correct about satellites? All right, how did we do? So the right answer is D. In plants, they cause symptoms distinct from helper virus. So the statement, there are no human satellites, is wrong. Delta is a satellite, right? They're defective viruses derived from the helper. I said that on one side. They're not defective viruses derived from the helper. Some of them encode protein, which is good. No one's picked that. All of the above are correct. Is not right, as I just told you. So they cause symptoms in plants distinct from the helper virus alone. Now we're going to talk about virophages. So this word means virus eater. It's derived from bacteriophage. A bacteriophage means bacteria eater. Phagein is Greek to eat. And virophage means eats virus, which is a totally dumb name in my opinion, but it's there. It's not going to go away. These are circular double-stranded DNA viruses with an icosahedral capsid, and they will replicate only in cells infected with a giant virus, big virus, not within the virus. So the press has somehow gotten the idea that this is a virus that replicates within another virus. That's not right. They replicate in a virus-infected cell, so it's an example of a helper satellite replication scheme, if you will, but they're not called satellites, they're called virophages. So they're globally distributed, and they have different names. One of them is called Sputnik. It was isolated from a cooling tower in Paris, France, and its helper virus is Mimi virus, which is a giant virus. And there's another one called Ma virus, whose helper is Cafeteria rhombogensis virus, etc. And so they've been found globally. This map below here shows all the places in the ocean and on land where we have isolated virophages, which again are simply viruses that need a helper virus, which happens to be a giant virus. That's how they are different. And there's the structure of one of these on the right. You can see it's a bona fide virus particle. And in cells that are infected with Mimi virus and the virophage, they disrupt Mimi virus replication. They actually reduce levels of Mimi virus production. So the Mimi virus are these particles with big icosahedral capsids, and they have kind of a fuzzy, hairy appearance around the outside. And these cells are co-infected with a virophage, Sputnik virophage in the case of Mimi virus. And you can see here that on the top, panel B, there's no DNA in this particle. It's actually full of little Sputnik particles. And then here's another one showing that. And then on the bottom here, you can see the morphological aberration induced by the Sputnik here, and another one on the bottom. So you make a lot of defective particles. Here's another one here in panel F. And you actually reduce the titer of the giant virus. So it's a weird arrangement where the virophage needs a helper, but it also inhibits the helper. It's like nothing like being grateful for what you're being provided, right? So are they satellites? People used to call these satellites, but they're not really. They are dependent on transcriptional machinery. So viruses need transcriptional machinery of the host cell, right? Especially the smaller ones. These viruses, these virophages also need a transcriptional machinery, but it happens to be virus transcriptional machinery, not that of the host. So, we don't call viruses satellites because they depend on the host. Call them viruses. So it's been argued that these should be simply called viruses, not satellites, because they depend on the transcriptional activity of another virus. And the problem is that satellites have been around for years, and so when these were discovered, they were automatically called satellites, but it's probably not right. They're just viruses or virophages. Okay, now we're going to talk about prions the rest of the time. And these are really interesting. So prions are infectious proteins with no nucleic acid component. And I found that picture of a road sign. I don't know where it is. It's called Prion Road. So prion is a name made up by Stanley Prusiner, who didn't discover them, but he showed how they worked, and he provided evidence for their existence. And these cause diseases that are always in the news, like mad cow disease, CJD, Scrapie, chronic wasting disease of deer and elk more recently. And if you are into ballet, you may know of George Balanchine, who used to be the director of the New York City Ballet years ago. And he died of a prion infection. And this is just an article from the Times where they talk about how his brain cells were examined after he died at 79. And they saw that he had had a prion disease. No one knew at the time what he died of, but post-mortem examination of his brain showed that he had a prion disease. And so the diseases caused by prions are called TSEs, transmissible spongiform encephalopathies. So an encephalopathy is a disease of the brain, and the transmissible, you understand what that means. It goes from one organism to another. And spongiform is the pattern of the brain that you see when you look at it under the microscope. And I'll show you that in a moment. These are fatal neurodegenerative disorders of mammals. They're fatal, uniformly fatal. We have no cure yet. If you are diagnosed with a prion or a TSE, you have no chance of surviving. And thousands of humans get this every year. Maybe 1% of them or by infection, and the rest are by other means, which I'll tell you in a moment. And we had an unfortunate incident in history when humans got a certain kind of TSE by eating cow meat that was contaminated with bovine spongiform encephalopathy prions. We'll talk about that. But these prions, basically, you cannot destroy them by cooking. You cannot destroy them by autoclaving or low pH or high pH or bleach. They're pretty scary. You can have your hamburger well done. It could still have a prion in it. So let's talk a little bit about the history. So there are TSEs of many mammals besides humans. There's BSE, which is also called mad cow disease. There's chronic wasting disease of deer, elk, and moose. There's exotic ungulate encephalopathy. There's feline prion diseases. So cat lovers, your cats could get a prion disease. Also, lions and tigers can get them, the great cats. There's scrapie in sheep and goats, and even minks can get prion diseases. So they're all over the place. And we have a bunch of them in humans. They have different names because they're slightly different diseases. We have Creutzfeldt-Jakob. We have fatal familial insomnia, Gerstmann-Straussler, Kuru, and variant CJ. Variant CJ is what we get from eating cow meat that has bovine spongiform encephalopathy prions in it. And so spongiform comes from examination of brain slices. You see that has holes. It looks like a sponge. It's the brain. It's not supposed to look like that. There's just supposed to be holes in your brain. That's bad. And so you have, throughout the brain of a person with a TSE, you have these spongiform appearances. You have motor dysfunction. You have cognitive dysfunction. Many of these individuals stop talking. They just sit and stare, and that's because of the brain damage here. And each disease has a slightly different kind of symptomatology. The different human diseases I told you about before, slightly different symptomatology and pathology depending on where the brain is actually infected. So the first TSE ever recognized was Scrapie. And this is, it's called Scrapie because the sheep with this disease tend to rub themselves on fences. And so that sheep, for example, has Scrapie, and a lot of its fur is rubbed off in certain areas, as you can see by rubbing against the fence. These sheep have motor disturbances. They have uncontrollable trembling. In Canada, it was called tremblement de mouton. Paralysis, weight loss, and usually 4 to 6 weeks after seeing the first symptoms, the sheep die. And this has been known in European sheep for a long time, over 250 years, and it's currently still endemic in many countries. For example, 1% of sheep every year in the UK come down with Scrapie. All right, so that was the first one. And people started studying this to understand what it was. And farmers made the first observations. They found that it could be transmitted. If, say, a farmer gave a sheep to his neighbor, it turned out, and that sheep had Scrapie, but the farmer didn't know it, then the neighbor's sheep would get Scrapie. So it seemed to be transmissible. And so scientists began working on it, and they found that if you take the brains of sheep with Scrapie, you can grind it up and inject it into another sheep and they will get Scrapie. But the agent passes through a filter. So for many years, people thought it might be a virus. However, the Scrapie agent is not inactivated by ultraviolet light or ionizing radiation or formaldehyde or autoclaving or any of those things that would inactivate a virus. And so very early on, it was thought there's no nucleic acid in this agent, whatever it is. This is one of the early experiments that was done, 1984, to show that Scrapie is really different from other viruses. So this is an activation curve. So it's the dose of irradiation that you need to give to get an activation. You get an activation curves, and here is the size of the genome in kilobases and molecular weight. So basically, these are inactivation curves with increasing amounts of radiation. And you can see that some viruses are inactivated with pretty low doses, like herpes and vaccinia, big DNA phages. And then as the genome gets smaller, so SV40, really small, polyoma, you have to use more radiation to inactivate it because the target is smaller, right? It's less likely to be hit by radiation. But look at the Scrapie radiation is way at the end here. You need a lot of radiation, huge amounts of radiation to inactivate infectivity. These are RNA viruses here, same pattern. The big ones, you don't need a lot of radiation. And so Scrapie is needs more radioactivity or radiation, irradiation to inactivate it than every virus, except the circoviruses with the circular DNA genomes. So that was a clue that this was a unique agent. Early on, it was recognized that the animal TSEs and the human ones share a lot of properties, and that therefore we could study TSEs in animal models and learn about what's happening in humans. So there's plasma membrane defects, there's vacuolation of neurons and astrocytes, the oligodendrocytes, the spongiform appearance, loss of neurons, accumulation of glial fibrillary acidic protein in clumps in the brain, amyloidosis, fibrils of misfolded prion protein, a lot of characteristics shared with other human degenerative neurological diseases, but also some unique properties. So people started studying Scrapie to understand them. And so they found that you can make homogenates of organs and inject them into another animal, and they will cause the same kinds of symptoms, ataxia, dementia, death after many years. When you do this, the agent first accumulates in the lymph system and secretory organs and spreads to the central nervous system where you get the damage happening there. And interestingly, there's no inflammatory response, right? When you put a virus into a host, you get inflammation as the virus reproduces. There's no inflammation. There's no adaptive response. There's no antibody response. There's no cellular response to the prion. And that's because it's a self-protein, as you'll see in a bit. And these are, as I said, invariably fatal. You never know who has it until they begin to develop the symptoms. Like, typically the first ones are having altered mobility, difficulty in walking, then you have cognitive symptoms and so forth. They can't be treated. There's no way to alleviate symptoms, so they're all invariably fatal. We have no way, even to this day, we don't have anything with which to treat these diseases. Now, in 1967, a mathematician by the name of Griffith said, I think these are proteins. And so that got Stan Prusner interested many years later. He was a neurologist working at UCSF, and he decided to work on these. He had seen some people with prion diseases, and he started working on Scrapie. And he found that there were proteins in the Scrapie brain which he could purify and then inject into animals. He used hamsters and then mice, and they would develop a Scrapie-like disease. He called the agent a prion, proteinaceous and infectious particle. So he made up that name, and it's stuck, of course, but it's kind of a neat name. The prion protein, so this is what he purified, a protein from Scrapie, infected, Scrapie suffering sheep. He got a single protein which he could inject into other hosts and it would cause the disease. He found this is encoded by a single gene in mammals called the PRNP gene. And you need that gene to develop a TSE. How do we know that? Well, we can knock it out in mice, and if you inject prions into them, they never develop a prion disease. So the PRNP gene encodes the prion protein, and it's essential for the pathogenesis. And our current view of what's going on here is that the prion protein that causes disease is a misfolded form of a normal host protein. So the normal host protein is called PRPC for cellular. It's normally on the outer surface of neurons. It's anchored by a GPI linkage. It's not a transmembrane protein. And then somehow it misfolds. And the misfolded protein leads to the prion, the TSE disease. But even more than that, you're full of a normal folded prion protein. When you encounter some misfolded prion protein, it causes the normal protein to misfold. And so you get more and more of the misfolded protein produced, and that leads to the more and more severe symptoms of the disease. What's the point of having a gene that will give you bad disease, right? Yeah. I mean, I don't know the answer because you can knock it out in mice and they seem to be fine. Yeah. So it may have a redundant function. There may be other genes that substitute for it, but we have the gene as well. And so it's doing something that we don't understand. Yeah. All right. So here is our view of prions and how we can actually detect them. So the prion protein is shown here, PRPC. It's got a certain structure with alpha helices. The structure of it's actually been solved, and it has a couple of beta strands there, and then 2 alpha helices, right? And if you add a protease, an enzyme called proteinase K, it will completely digest this protein. So people early on made antibodies to this, and they could do Western blots and look at the normal prion protein. It would be degraded in the presence of proteinase K. When it misfolds and becomes PRPSC, so SC is for Scrapie, just to designate a misfolded form, it now has a lot of beta sheet conformation. You can see it goes from having 2 beta strands to having many. And this protein is now resistant to proteinase K. If you digest it, it only takes away the N-terminus, and the rest is a 27 to 30 kilodalton form, which you can detect on a Western blot. And so this is one way you can tell if someone has Scrapie, TSE, a human anyway. You do a Western blot of their brain, which you have to do after they die, right? You can't do it beforehand. And you do a Western blot and you probe with antibodies, and you can see the production of this altered form. Nowadays, we can actually do some other tests, which I'll tell you about in a bit. So the key experiment, in my opinion, was showing that if you knock out both copies of the PRNP gene in mice, they're resistant to infection. They will not get a prion disease. So you can give mice Scrapie prions. If they're lacking the PRNP gene, they will not develop Scrapie. And the way you acquire TSEs, you can get them by infection. You can eat meat that's contaminated with a prion, or there are other methods as well. Or they can be spontaneous mutations occurring in the PRNP gene that cause it to misfold. There's two different ways. And eventually, whether you acquire it by infection or it misfolds, it accumulates in you, the misfolded protein accumulates, and eventually you get the symptoms leading to the disease. And there are three types of these TSEs. There's the infectious kind, shown at the top there. So you somehow get some PRP, misfolded PRPSC. And that comes in you and makes your PRPC misfold, and then you get more and more PRPSC, and then eventually you get the disease. So infectious, you could eat it, for example, or you can get it in a blood transfusion or tissue transplants and so forth. There's a genetic form, or familial, where you inherit mutations in the PRNP gene from your parents, and they cause the protein to spontaneously misfold and eventually amplify the misfolded protein. And then there's what's called the sporadic form, where you haven't eaten anything that's contaminated, you don't have any mutations in the PRNP gene, but the protein just spontaneously misfolds. And once you have a little bit of PRPSC, misfolded PRP, then you develop a TSE. And all of these can be transmitted to animals by inoculation or by feeding them infected tissue. So even though they arise in different ways, in the end, the PRPSC is infectious. So those are the three kinds there, the infectious kind, the genetic kind, and the sporadic kind. So one kind of infectious human TSE is called Kuru. Talk to you about that in a moment. We can also transmit PRPSC by infections by transplantation of infected corneas, by using hormones that we purify from people, by blood transfusion. So before we understood the origin of TSEs, we would give people blood transfusions and corneal transplants and hormones produced from people, and we inadvertently gave them TSEs because we didn't know what was there. And then once we found out, well, for example, ophthalmologists would do procedures on patients where they would touch tools to the eye, and they would get contaminated with prions, and they autoclave these between patients, but autoclaving does not destroy infectivity. So a lot of ophthalmological procedures transmitted TSEs to patients. BSE was an example of, so BSE was a prion disease in cattle, which happened because we were feeding cows sheep who had died of Scrapie. And then we ate the infected cows, and we developed variant CJD by eating BSE beef. So those are all infectious forms. Let me tell you a little bit more about Kuru. Kuru was a fatal TSE found in the four people of New Guinea here. Here it is in the Pacific. And long incubation period. Carlton Gajicek, a virologist in the US, went to study this disease. And he found that it only affected women and children, because when someone in this four people died, the ritual of the funeral was for the women and children to eat some of the brain of the deceased person. And so you can imagine that if someone died of sporadic TSE, then their brain is eaten, you're going to transmit it to the women and children, and they're going to die, and you're going to eat their, so it kept going on and on. And so he told them, you can't eat the brains anymore. They stopped, and there was no more Kuru now. So it's a disease of cannibalism. Sporadic CJ, where there's no infection and no genetic component, it affects 1 to 2 per million humans worldwide. So it's a lot of cases. They're typically for 50 to 70 years of age. That accounts for 65% of all the TSEs in humans. So the sporadic form, where there's no eating or contamination or infection, and there's no genetic predisposition. These patients have normal PRNP genes, but they develop sporadic TSE can be transmitted to other people via transplants and so forth. And we think this is what's established Kuru in New Guinea by eating the brain of a person with sporadic CJD. And then there's familial, where you have mutations you inherit in the PRNP gene from your parents. It's an autosomal dominant mutation. There are multiple such mutations. And again, when you develop this, you can transmit it to others by transplants, of various sorts. So here's a graph of CJD. This is one kind of TSE in humans, Creutzfeldt-Jakob disease. In the US, the deaths are in blue, and the age-related death rate is in red. So the number of deaths on the y-axis here, and then deaths per million people on the right. So you can see increasing numbers of people dying, and so 500 here at the peak. But the death rate, the age-adjusted death rate is more or less constant. So how does this work when you eat something that is contaminated? Well, this picture actually tries to summarize all three ways of getting a TSE: the genetic, where you have a mutation in PRNP; the infected, where you eat something or you're given a transplant with contaminated PRPSC; and then the sporadic. The sporadic and the genetic, you get misfolding in the brain. But if you eat meat that contains prions, like BSC, contaminated meat, we think that the prions go into your intestine. They cause prions to misfold on the intestinal epithelium, and then those make their way to the CNS by exonal transport. Now, in the UK, there was an epidemic of a TSE called bovine spongiform encephalopathy among British cattle. This is a form of cannibalism. That's a cow with, end-stage TSE. It can no longer walk. And this is from feeding sheep meat to cattle. So if you feed cattle protein, they grow faster. And so the people always have fed protein to cattle to do that, but in the 1970s in the UK, they changed how the meat was prepared. So you typically would get animal carcasses and prepared a certain way. In the 1970s, they changed how the meat was prepared, and this now allowed the Scrapie prions to pass from the feed into the cows. And we recognized this increase in mad cow disease. We stopped preparing the meat this way, and then the amount of BSE decreased. And then people who ate this contaminated meat, because for many years we didn't know about it, so the cow meat got into the food chain. They developed variant CJD, and some of them passed it on to other humans. So here's the epidemiological curve of this outbreak. Here's the BSE outbreak starting in the late 80s and peaking, and then going down when we changed the processing of the food, and then the overlapping human outbreak of variant CJD, which eventually went down. And then some cases of blood transmission of these patients with variant CJ, giving it to other patients. So we had about 1 to 2 million cattle in the UK infected with prions during this time. And the main problem is with cows is that you don't see symptoms until 5 years of incubation, and most cows are slaughtered at 2 to 3 years of age for meat production. And so that's what got the prions into the human food chain. We still have cases of BSE in cattle. Those are probably sporadic, where a cow will just develop a sporadic BSE. And so we're trying to deal with this, but we don't test much of our cows, right? We have a huge cow industry, not just for milk, but for beef. We test less than 2% of cows for prions. And so we've tried to develop diagnostic tests. We're trying to develop drugs that would deal with this, but we don't have any drugs that would inhibit prion production. But we do have diagnostic tests. These are the cases of BSE in North America from 93 to 2018, the USA and Canada. You can see there are not a lot of cases. There are a total of 26. But if any one of these gets into the food chain undetected, it could cause an outbreak of variant CJD in these countries. And I have here 2 articles I wrote about prion disease being acquired from from different sources. So the top is prion disease from contaminated beef. This is a gentleman who was living in, I think, Saudi Arabia and died of what was a TSE. And it turned out he had been living in the UK during the time of the BSE outbreak, and he had consumed beef. So these people move elsewhere and they develop disease. In fact, if you lived in the UK in that time of the BSE outbreak, you are not allowed to donate blood for the rest of your life, because the possibility is you may have misfolded prions in you. And then the second article is very interesting. This is prion contamination in an ER. This is a case of a man who was demented. He walked into an ER and shot himself. He killed himself by shooting himself in the brain, and his brains splattered all over the ER. And they found out later that he had a TSE, which was causing his dementia, and they had to throw out all of the ER equipment, hundreds of thousands of dollars, because you can't sterilize it. There's no way to remove the prions from the equipment. So every ER now should have a protocol in place if someone comes in acting like this, to get them isolated before they can do that. I interviewed Richard Knight a couple of years ago, who's an expert in the UK on prion diseases, and that's an episode of TWIV you might want to listen to. So here are 3 epidemic curves for Creutzfeldt-Jakob disease. And the top one is showing you the number of infected patients with time. This is the outbreak of Creutzfeldt-Jakob caused by giving people growth hormones that were extracted from human pituitary glands. So nowadays we make recombinant growth hormone for people who need it. But in the old days, we used to take pituitaries from deceased people and extract the protein and inject it into people. And there was an outbreak of a TSE caused by that procedure, which you can see peaked in the late 90s. There's also an outbreak of CJ caused by transplantation of dura mater grafts, so pieces of neurological tissue transplanted into people unknowingly from people who had early stage prion diseases. And then at the bottom is the variant CJ outbreak from eating BSE contaminated beef. So 3 outbreaks of this, all caused by infectious roots. There is a prion species barrier, and that is that in most cases you have to put the misfolded protein in the same species where it originated to get disease. If you put it in a different host, usually disease is inefficient. And so the misfolded prion sequence has to match in both the donor and the recipient. So the sequence of the prion protein determines the tropism, if you will. So here are 2 examples. If you take a misfolded hamster prion, hamster PRPSC, and you inject it into mice, there's no disease because the protein is a different sequence in hamsters and in mice. But if you make mice transgenic for the hamster gene, then you inject those mice with hamster PRPSC, then the mice will develop disease. So it's a nice illustration of how not only do you need the PRNP gene, but it has to match the organism that it's coming from. If you put, however, bovine PRPSC into mice, they do get disease because apparently the cow PRPSC has a broad host range and infects many mammals, not just mice. So somehow some prions overcome this host barrier. And that's why we're worried about BSE, because it not only infects humans, but it's been given to zoo animals by feeding them contaminated beef. All right, our last question is, which of the following observations illustrate the prion species barrier? A, mice lacking the PRNP gene do not develop disease. Mice fed hamster PRPSC do not develop disease unless they have a hamster PRNP transgene. Humans consuming BSE beef develop disease. Humans may sporadically develop prion disease, or humans may transmit prion disease to other humans via corneal transmission. So which illustrates the species barrier? Let's have a look. 9/11. Yes, mice fed hamster prions do not develop disease unless they have a hamster PRNP transgene. That illustrates the species barrier. Mice lacking the gene do not develop disease. That's that's all about saying that the PRNP gene is important for disease. Humans consuming BSC develop disease. That doesn't illustrate the barrier. Humans may sporadically develop, doesn't illustrate the barrier. Humans may transmit by corneal transplants. It doesn't illustrate the barrier. They're all true observations, but it doesn't illustrate the species barrier. Now, the latest concern is a disease, a prion disease called chronic wasting disease, and this is a prion disease of deer and elk and moose. They're called cervids. It's been detected throughout the US and Canada in both free-ranging cervids and in herds. And in these, the very high percentage of animals can be positive, and in the wild, up to 15%. So on the left is a graph of the number of states in the US with reported cases with time. And here you can see all the states in the US and Canada that are reporting cases of chronic wasting disease in either free-ranging or captive populations. And this is a concern because of several reasons. First of all, deer and so forth excrete urine and feces in the wild, and it's been shown that plants can take up prions from the soil. They can be present on the leaves, and if animals take them, they can get infected. So we're worried that, say, a deer will contaminate a pasture, and then the cows will come and eat the prions and develop a TSE, and then the humans will eat the cows and it will pass on. Now, if you put the cervid PRPSC, so you take from the deer the misfolded protein, you put it into mice, you don't get disease. If you put the cervid PRNP gene in mice, then you do get disease. So great, that matches. It's got a species barrier. It depends on the right gene. If you make mice transgenic for human PRNP and inject the cervid PRNP, you don't get any disease. So this suggests that humans should not be able to get the disease, but we're still concerned because more and more deer are being infected, and we're worried that maybe the prion will change slightly over time and be able to infect people. And so if you infect deer prion into raccoons, voles, and cows, intracerebrally, you get disease. Now, that's not a natural inoculation, but it suggests that that overcomes the species barriers in those individuals. So we're just worried that the deer will transmit the prions to other animals and eventually make its way into the food chain. So people are very concerned about this, and they're being monitored very closely. And hunters who hunt deer are told to be very careful with the deer carcass. Don't shoot a deer that's acting weird, first of all. Wear gloves when you take the deer apart. Don't get near the spinal cord and brain and so forth. Just worried. There have been no cases of humans getting a TSE from deer so far, but, it's a long incubation period. And I just want to end up with this picture, which kind of tries to tie all of this together. So, Scrapie was first identified many, many years ago. And that's a naturally occurring disease of sheep. And maybe chronic wasting disease is a naturally occurring disease of elk. In other words, sporadic misfolding or genetically based misfolding. And then these animals transmit it to others, right? We feed sheep to minks and they can get it. Perhaps this happened in the wild. When a mink fed on a sheep, we transmitted Scrapie to cows, who then passed it on to other cows, and they were fed to animals in zoos, and it was passed on to them, even cats, and then passed on to humans as well. It could be also that Creutzfeldt-Jakob arose in the 1920s as a sporadic disease of humans and was passed on by iatrogenic means, in other words, transplantations of corneas and different blood products. So there's a complicated relationship among these TSEs, and we're still trying to figure out, as I said, we have no way of treating these infections, and that's really a major goal. So next time for our last lecture, we'll talk about therapeutic viruses. So this takes everything you have learned, and we make viruses to treat diseases, to cure cancers, and to act as vaccines. So it's a really nice way to end this course.